So in addition to providing these little highways of transport, the cytoskeleton actually has a ton more functions in supporting the cell and organizing what's on the inside of the cell as well, right? So what we're going to be talking about here are the three different components of the cytoskeleton. Microtubules, they're the biggest ones. Microfilaments, they're the smallest ones as far as diameter goes. And then intermediate filaments, which are kind of in the middle. So microtubules, these are the really, really big ones as far as the diameter goes. You can see the little inset picture here. It's kind of a column of tubulin, and tubulin is just a protein that makes up these structures. Um, I would say probably the most important aspect of all of this. Now, obviously, I'm not going to have you guys remember how wide they are by the numbers. Should know that they're composed of protein, but it doesn't really matter to me if you know if it's tubulin, alpha and beta tubulin or not. But what I do think you should know is that it is really important in getting cells to move. That's with cilia and flagella. And it's also important in moving things around. That includes chromosomes as well as moving the organelles, those little functional entities inside the cell. So if you look at this, okay, these are microtubules, which are kind of short, and they arrange themselves in the cell so that they can start to produce even and serve as anchor points for even more filaments. So microtubules grow out from what's called a centrosome, right? So these microtubules, remember the thickest ones, right? They grow out from the centrosome, which are these organs here. You have one sideways here and, kind of one up and down. The centrosome is a microtubule organizing center. So in other words, instead of having a haphazard arrangement of these microtubules inside the cell, they're actually pretty orderly. So in animal cells, the centrosome has a pair of centrioles. Okay, so don't get confused between the centrosome and centrioles. Centrioles make up the centrosome. Okay, so in other words, the centrosome is really kind of like the point where they occur, in other words, right? These two structures occur. Um, and the way that this is arranged is what we call the 9 by 3 arrangement. So if you look at these, I mean, you could count them if you really wanted to. You can see three little tubes, right, of tubulin here, right? So there's three arranged in, if you go around the radius, there's nine, sets of three, which is why they call it that. Okay, so I think that's important actually because I always try to give you guys insight on, these standardized tests. If you take the HESIs or the MCATs or the biology GREs or something like that, they love this question too, that 9 by 3 arrangement, and this is how the tubules set themselves up with basically 27 if you multiply them, right? Tubulin fibers or filaments. So microtubules control the beating of cilia and flagella, which we sort of mentioned a little bit before. Cilia is like this little tiny carpet of hairs, of these tubulin filaments that come out. And, if you have a cell, you might have a whole bunch of these little tiny hairs covering the surface, right? So this might be, a pond organism or, just something, more like microorganisms. All these little tiny hairs that stick out of the outside of the cell will literally kind of like beat the water, almost kind of like swim fins, really, and that propels the cell forward. So that's what cilia is. Now, flagella, I think we're all probably familiar with this. Flagella is basically, the tail of a sperm cell, right? So um, that is one very, very long arrangement of tubulin, which allows movement for these cells through the environment. Now, the other thing you can do with these, though, is if you have a cell that is anchored in place, so say like a cell in your lung or something, they don't move around, they basically just stay there. If you have little cilia, say that these are, I don't know, we'll just draw four lung cells, little nucleus inside, right? They're all stacked together. If you have cilia on the outside, what these little cilia can do when they move is not move the cells because they're attached in place. They can move the liquid around the cell, So instead of, we use the swim fin analogy for these guys and they're moving through the environment. Well, what if you still have water and you still have swim fins, but you hold on to the edge of the pool, right? You're not moving yourself through the pool, but you are moving the water around you, right? And what that does is it helps clean the cell surfaces. It distributes materials in the lungs. There's all kinds of good stuff that happens when you just move the fluid around the cells itself. Okay, so Um, how this actually works is through a motor protein called dynein. Think like a dynamo or dynamic, right? That's what gets it to move. This drives the bending movements of a cilium or a flagellum. Okay, I'm actually going to show you a little sort of like demonstration of that in a second. Okay, so before I do that demonstration, let me just talk about the structure again. So you can see that sort of like 9 by 3 arrangement here, right? Or you can also have a doublet, where you have two of them that are arranged around this sort of like spoke, right? So, this little cilia, if you were to cut them through, this is what you'd see, right? So you'd see a little bit different arrangement at the base of the cilium. So you can see right where it goes into the cell membrane down here versus a little bit further down, towards the end of the cilium, it looks a little bit different. Okay, so keep in mind, if as far as cilia goes, they're all still wrapped up with the plasma membrane, which is like this little blue sleeve that goes around the outside. Now, how this actually works is you have, sets of filaments. Here you have your doublets, right? And then this red stuff in the middle is what's called dynein, right? So dynein is that sort of motor protein that's actually getting things to move. Like, think of the word dynamic, right? When you apply ATP, which is an energy source, to this protein, what it's going to do is get the filaments to slide relative to each other. And they're connected at the end, which is going to bend the tubule. Now, I really don't know if I can actually, explain this, with just this drawing, okay? So by sliding one end up, and keeping one end stationary, it basically bends the whole column of all of these proteins. Okay, so what I'm going to do for you is a magic trick, actually, and film myself. Man, I'm really throwing myself out here for this one. But this is how I generally explain it in class. So hopefully this will make sense to you here. Keep in mind what you want to know, or really the end result here, is that these little arms of protein, these little filaments of protein will actually, wag back and forth. And what that does is it basically moves the fluid around the cell, right? So we're talking about very, very tiny stuff here. Okay, so here, let me try this and see if this actually works. Hey everybody. All right, so what I think I'm going to try to do here is to use this, right? So this is just a piece of paper. It's folded in the middle. It's got the corner on the end, so you can just basically see what it looks like here. And what I'm going to do is pass an imaginary needle through it. Yeah, I said imaginary. All right, ready? So I'm going to hold this in my fingers, and I have this a needle. It's invisible. You can't see it. And if I push this through the paper, what's going to happen is, I'm going to grab it the other side. Okay, is that it's actually going to bend the paper, right? Now, if I let it go, it should go back to normal again. Okay, so you see what's going on here? Just kidding, no imaginary needle. All I'm doing here is using my fingers, okay, to slide one side up and keep the other stationary, right? So what that does is, and as you can see here, right, what that does, put against my dark shirt, is to wiggle one way and then wiggle the other way, right? So if you think about it, it's not a perfect example here, but if you think about it, as these little cilia are moving around on the surface of the cell, it's basically doing this, okay? With that little dynein arm would be in this space in the middle here, okay? So that's how it works. With a little bit of energy, you're getting a cell, imagine this is the tail of a sperm or something like that, just swimming through the environment just by moving these things back and forth, okay? So hopefully that kind of like pulls together what all these crazy pictures are. I don't think if I was a student I could ever really get what was going on there without actually seeing it in action. So since we're not in class, I figured I'd just video myself for this one. Okay, so any questions, please let me know. Okay, so as I just showed you, again, not a perfect example, but that's basically what's going on here. The pattern of movement's a little bit different. Flagella on the end of a sperm is actually almost kind of like a rotation, like think more like a propeller than a back and forth. But I think everybody probably gets the idea, unless you're working in fertility studies, with sperm specifically, or, you're a microbiologist. Hey, it'd be great if you were, if that's what you want to do. But I think we're just going to leave it at that as far as, how this actually works, right? So, as you can see, okay, the cilia, will basically go back and forth. And what that leads to is, the organism going in, well, to the right in this case, right? So the power stroke versus the recovery stroke, or what they're calling the active stroke instead of the power stroke here, right? Pushes backwards, that sends the organism forwards, and then there's a recovery. Think about how a bird flaps its wings, right? It pushes down hard if it wants to gain altitude, right? Pushes downward hard, and then pulls its wings in so that there's not as much resistance as it's bringing its wings back up again, right? And then pushes down hard again. That gets the bird to rise up into the air. That's basically what you're seeing here. Okay, so microfilaments. Okay, so these are the smallest ones anyway. And I would say, as far as what you need to know for this, don't worry about all this craziness. Know that it is a different protein. It's called actin, and it almost kind of looks like a rope over here, right? Um, and what it does is it bears tension in cells. Okay, so in other words, if you're getting a cell and pulling on the ends, it resists that pull so that you don't tear the cell in half. Okay, now think about that. That's exactly how your muscles work, right? So that's why we're calling it muscle contraction. Also changes the cell shape as well, right? So that's the big one I think that you should know. There's also a little bit of moving around inside the cell, moving the cell through its environment, which is what we call motility. Okay, it causes the cell to sort of like tumble through its environment, or on a surface, and that's what gets movement to happen. And then for cell division, it'll lead to what's called a cleavage furrow. Okay, so again, if it's been a while since high school biology, you got a cell that's ready to divide. It'll start to kind of like pinch in the middle, right? And then it'll eventually form two cells. That's what we call the cleavage furrow, right? So a lot of functions for this one. So these microfilaments are, solid rods and they're twisted as I just showed you, right? But they form a 3D network, so it's kind of like a playground where you have not just a rope, but ropes going all different directions, or sometimes they're actually organized in the same direction depending on what the cells want to do. Your muscle cells are all pretty much in the same direction, right? So that's, I think, important. And also, kind of along with muscles, they also make up, what are called microvilli. Okay, these are little surfaces or extensions of surfaces on cells that add to their surface area, and that's what you're seeing here. So you got these little microvilli, which have the plasma membrane going around them. You can see how they're rooted, and you can see how crazy this network is. That was the playground sort of arrangement I showed you before with the ropes, or told you before, with the ropes going in all different directions. That's just adding to structure of the cell. Okay. Here, what you see, I'll get rid of this stuff. Here, what you see are muscle cells. So this long cylinder right here is one cell. And what happens is you can see the arrangements of filaments, right? So there's the actin. There's another type of filament in your muscles called myosin, which we can talk about. But what these do, see how they're all arranged in parallel lines? They'll pull inwards towards their centers, and that's what actually gets muscles to shorten. So if you think about it, flex your bicep or something like that. Really, what's going on is that muscle is shortening the distance between the bones in your forearm, namely your radius, and your shoulder blade, right, which is also your scapula, which is, behind your shoulder. That's what your bicep is attached to. So when you shorten that bicep, think about, you're just flexing your guns out, right? You're going to pull those two bones closer together. And that's how you move, right? All 640 plus muscles in the body work this way. So I would say that, knowing the muscle example is probably what I'll be referring to. But cells also use these little, filaments of actin, or microfilaments. They basically stream them all in one direction, and that gets the cell to basically kind of like, I don't, lack of a better word, kind of like blob towards, the left in this case. And that little new extension of cell material is called a pseudopodium. So a pseudopod, is basically, it literally translates to false foot. So it almost kind of looks like the cell is like, moving a foot forward in order to move. Okay, so that's going to do it for this right here. I think we'll probably get one more video out of